Hello. In this module, we're going to talk about sex. But what exactly do I mean when I say sex? What first comes to your mind? Copulation, women and men, gender roles, anatomy, genitals, sexual orientation, reproduction, fertility. Sex is such a broad topic and is also a really interesting topic for a lot of people. Of course it is. Sexual reproduction started about 2 billion years ago when the earth was occupied by just little protists. Initially, single-celled organisms, they just cloned themselves. But once the ability to reproduce sexually became a thing that could actually happen, the advantage to doing so experienced significant and positive selection. Now, a big advantage to sexual reproduction compared to cloning is that genetic variation is introduced by the recombination of 2 genomes. So this recombination of genomes has proven to be an evolutionarily winning strategy, especially when a species needs to adapt to a changing environment. In species that can reproduce sexually and asexually, meaning that they can also clone themselves, biologists have found that when these organisms are pretty well suited to their current environment, they'll reproduce asexually. But as soon as there's an environmental shift, they'll start to reproduce sexually. Now, sex is also a very useful strategy when there aren't all that many individuals in a population. This is because recombination helps to reveal genetic variation that's recessive or epistatic, meaning that the combination of 2 or more genes influences how expression occurs. So the more you shake things up from different combinations, the more variation will be made available for selection to operate on. So, sex has been going on for longer than metazoans. Animals have been around. Essentially, all animals can reproduce sexually. What this means then is that for over 1 billion years, genetic variation that leads individuals to be interested in sexual reproduction have been positively selected for. If you are interested in sex, that's because all of your ancestors for more than 1.3 billion years, they were interested in sex too. And because they were, that led them to be more successful in reproducing, and therefore they passed on their interested in sex alleles to their descendants. And consequently, their descendants were, and still are, interested in sex. Now, before we go much further, I want to take a moment to talk about how you engage with the next 2 modules. I know many of you may be living with family of different generations. Now, my granddad, he had some very strong opinions about, well, honestly, he had very strong opinions about everything. And of course, he had strong opinions about sex. And these opinions included the assertion that if I ever had anything to do with boys or sex before I was properly married, even if I just thought about those things before I was properly married, he'd throw me off the New River Gorge Bridge. This is the New River Gorge Bridge. I most definitely did not want to be thrown off this bridge. I remember quite a few Thanksgiving dinners looking at him in horror across the table as he would say this, just surprised at the vehemence in his tone. And my granddad, he's the guy, he loved to wrap presents with these beautifully hand-tied bows, and yet he'd throw me off the bridge. And I don't think he was kidding, honestly. Jeez. I share this story with you because if you live with a family member like my grandfather, who might be looking over your shoulder, you will probably want to find perhaps a more private way to watch these videos, or just listen to the audio. that will work too. We will focus first on sexual differentiation, how male and female are defined and how they develop. Sex differentiation, it's not a simple thing. There's no one magic switch that explains the distinction between male and female. We will divide the dimensions of the process of sexual differentiation into 4 main categories. So 1, genetics of sex, gonadal sex, and the morphology of the external genitalia, and also brain development. Now, these aspects of differentiation, they happen at different times during development. Genetic sex is set at fertilization. Gonadal sex is set at fertilization. Gonadal sex and the shape of the external genitalia form during the first trimester of pregnancy. And brain development, when sexual orientation and gender identity are largely determined, that occurs during the 2nd trimester. This module will cover all of these topics, but this video will focus on the first, genetic sex. I'm going to break it into 2 segments though, so that no one video is too terribly long. So speaking of genetic sex determination, we're going to cover 6 key topics. In the first video, I'll tell you about sex chromosomes versus autosomes, the X chromosome, dosage compensation, and X inactivation, and we'll talk about the pseudoautosomal region of the sex chromosomes. In the second video, jump through my animations here. In the second video, we will talk about the Y chromosome and genetic imprinting, and I'll give you a few examples of genetic imprinting by way of trying to help you understand how that works. Let's begin with sex chromosomes. You're already familiar with the distinction between the autosomes and the sex chromosomes. Humans have 23 pairs of autosomes, and then we have that 23rd pair of chromosomes, the sex chromosomes. This is typically a pair of 2 X chromosomes or 1 X and 1 Y chromosome, but not always. There are other viable combinations. In fact, out of every 500 babies born, 1 of them will not have an XX or an XY pairing of the sex chromosomes. Now, for some perspective, that means that right now, there are as many Berkeley undergraduates with sex chromosomes that are not XX or XY as there are students who play on Berkeley's intercollegiate athletics rugby team. Approximately 60. Now, you already know that mom passes along an X chromosome to her children, and dad passes on either an X or a Y. In the karyotype you see here, the X chromosome is the big one, and the Y is the tiny little chromosome just beside it. The X chromosome is actually the 8th largest of all the chromosomes in the human genome. Now, thanks to the Human Genome Project, we know quite a bit about its DNA sequence. The entire sequence of the human X chromosome was published in 2005. Now hold that thought. We'll come back to it in a few minutes. First, let's consider the fact that we have X and Y chromosomes, and that the presence or absence of the Y chromosome determines what a person's genetic sex is. This type of genetic sex determination, it's not universal. Not everybody does this. In fact, if you go back a little more than 300 million years, the differentiation between male and female individuals wasn't reliant on the genotype of the individual at all. Rather, sex determination looked like what it does today for this critter, the alligator. A mama alligator lays her eggs, and if the temperature is on the cold side or the hot side, all those eggs will hatch female alligators. If the temperature is in the middle, not extreme in either direction, all of the hatchlings will be male. This is what we call temperature-dependent sex determination. But it isn't actually an either-or situation all the time. Temperature-dependent sex determination versus genetic sex determination. While most reptiles have temperature-dependent sex determination, there are some that actually have a combination approach. For example, the Australian skink lizard has X and Y chromosomes that determine sex. But when the eggs incubate in low temperatures, there will be males who are both XX and XY. Wow! Evolutionary biologists think that some situation like the Australian skink lizard is likely what the earliest genetic sex determination looked like. Now, it turns out that when you survey across all animals alive today, genetically determined sex evolved multiple times. In reptiles, sometimes it's the males that are XY, and sometimes the males are ZZ. What? Let me introduce you to a new term, heterogametic. We are familiar with mammalian females having 2 X chromosomes and males having an X and a Y. The males are therefore the heterogametic sex. Females are homogametic. But it is the opposite in birds and some reptiles, as I just mentioned. The male is ZZ and the female is ZW, the heterogametic sex. Let's think about what it means to be heterogametic for a few minutes. And let's focus on mammals. So we'll think about the X chromosomes, and then we'll think about the Y chromosomes. The X and Y chromosomes, way back 300 million years ago, were just another pair of autosomes. Over that time, the X chromosome has lost almost all traces of that ancestral autosome. But the X chromosome is pretty impressively conserved. So just to make sure I said that the Y chromosome has lost almost all traces of that ancestral autosome. But the X chromosome is pretty impressively conserved, meaning that the X chromosomes of humans and mice and rats are very similar. Human and dog X chromosomes, they're actually even more similar than humans and mice. This tells us that the major characteristics of our X chromosome evolved before the radiation of mammals 105 million years ago. We've actually seen this before when in Module 4 I showed you this map. This map indicates chromosomal similarity between human and mice genomes. Now notice the X chromosomes, which are indicated in the color hot pink. In this map, at this scale, at this level of comparison, they are the same. Also, back in Module 4, when we first talked about autosomes and sex chromosomes, you were introduced to aneuploidies. An aneuploidy is when chromosomes are not in pairs. Sometimes during fertilization, there will be a third chromosome that gets encompassed into the gamete, the sperm or the egg cell, or sometimes one is lost, so chromosome goes it solo. The bigger the chromosome, the more unlikely it is that the fertilized egg will be viable. This is because the bigger the chromosome, generally speaking, the more genes on that chromosome. Now, embryos develop through a fine balance of gene expression. When a chromosome is missing or is in triplicate, that balance of gene expression, it's thrown off. For smaller chromosomes, that isn't such a big deal. And this is why people who have a third chromosome 21, for example, they not only survive, but they're beloved contributing members of families. These are people who have Down syndrome. So some chromosomes you are small enough and have a small enough effect on the phenotype that you can have an extra one, and still be fine. Now remember, I just told you that the X chromosome is the 8th largest chromosome in the human genome. There are over 1,000 genes on the X chromosome. Okay, now half the humans on Earth then have just one X chromosome, and the other half have two. So how in the world does that not lead to some serious dosage issues in terms of gene expression? Well, it works out through this totally cool phenomena where one of the X chromosomes in a female shuts itself off, inactivates itself. Very early in development, when an embryo is just 8 cells, and well before it implants in the uterus, each cell of a female mammal begins a process through which one of the X chromosomes inactivates itself. It turns itself into what we call a bar body. As to whether or not it's the maternal or the paternal X that shuts down during this stage of development, that appears to be entirely random. But what isn't random is that from there on, as the embryo continues to develop, all of the daughter cells that form from that initial cell have the same X chromosome inactivated, generally speaking. And the same X chromosome, have the same X chromosome activated and the same X chromosome inactivated, generally speaking. Mammalian males, including humans, they don't do this. Since they only have one X chromosome, every cell in their body has the same X chromosome that's working. But for females, there's variation. About 50% of their working X chromosomes came from mom, and the other 50% of working chromosomes came from their dad. Probably most biologists' favorite example of X inactivation is seen in cats. Now, one of the genes influencing coat color in cats is X-linked, meaning that it's found on the X chromosome. So in cats, the gene on the X chromosome influencing coat color is either an orange pigment allele or a black pigment allele. Since there is only one X in males, all of the cat's coat color will be the same, orange or black. Now, if a female kitty is homozygous, if she has two alleles for black fur, she'll have black fur. If she has 2 alleles for orange fur, she'll have orange fur. But if she is heterozygous, such that one X chromosome has the black fur allele and the other X chromosome has the orange fur allele, during the process of X inactivation, one of these will randomly be silenced, and the cat ends up with a patchwork of orange and black fur. This is how you get calico kitties. And this is why most calico kitties are female. If you ever run across a calico cat that is male, what do you immediately know about his genotype? Yes, he's unusual in that in order to have X inactivation, that means he has 2 X chromosomes in addition to his Y chromosome. A male calico cat has 3 sex chromosomes. He is XXY. This combination of chromosomes, this combination of sex chromosomes, is one of the most common variations seen in human sex chromosome variation. So you have the XX and the XY, which are the most common, but 1 in about 500 men is XXY. This is known as Klinefelter syndrome. The effects on the person's phenotype can be pretty mild for the most part, and often men don't even know they have it. But they often have some reduced fertility, they may be notably taller, maybe have poor coordination, less body hair, and a lower interest in sex compared to many other men. But the bonus is that they don't suffer from X-linked recessive disorders that affect other men. But I'm getting ahead of myself. One more thing about X inactivation before we move on to the next topic. Cloned cats. Okay, so in December of 2001, we had the world's first successfully cloned cat. Now, the donor cat is up in the upper left-hand corner, and her name is Rainbow, a calico cat, as you can see there, with spotches of white. And then the calico kitty over on the upper right-hand one, that is, the mama is the one who carried the donor embryo. So she's the surrogate. And notice the cute little gray and white kitten. That one is the cloned copy of Rainbow. And the scientist named her Cece for carbon copy. Maybe you all don't know what a carbon copy is because you've probably never typed on an old-fashioned typewriter. But in old-fashioned typewriters, you would take two pieces of paper and a piece of carbon paper you would put in between it. And then when you typed, you got the page that you were typing on, and then you also got a copy of it, a carbon copy behind it. So, CC, carbon copy, cloned cat. Anyway. But one of the things, as you look at that picture at the bottom of the screen, notice that you have Rainbow sniffing CC, and they don't look alike, do they? And they don't look alike because X inactivation in the fur coat is random. So anytime you clone a female cat, they're not going to be identical, in part because of the randomness of this type of X inactivation. As I mentioned a few minutes ago, the X chromosome has over 1,000 genes, about 1,098 of them, actually. This is about 4% of all the genes in the human genome, despite the fact that it contains only 4% of the genes, despite the fact that the X chromosome only contains about 4% of genes in the human genome. About 10% of the genetic diseases that we know of that have a Mendelian pattern of inheritance, 10% of those are due to genes on the X chromosome. They're what we call X-linked. So to be a bit more specific, about 307 genetic diseases out of over 3,000 identified to date are X-linked. And you're probably already aware of male problems that arise from having just one copy of X. And this means that if they inherit a misfunctioning gene on their X chromosome, they're pretty much stuck with the consequences. In females, the other X will be able to compensate. But for males, they don't have this genetic backup. Examples of these X-linked genetic diseases are hemophilia and color blindness. So 4% of all the genes, 10% of the diseases we've recognized with a Mendelian pattern of inheritance. So another question for you. What do you know about a woman's genotype if she's colorblind? I'm going to back up a sec. Oops. So what do you know about a woman's genotype if she's colorblind? So thinking about X-linked traits. Well, that's right. She either has only one X chromosome or she has a deletion on part of an X chromosome. Okay. Now what I want to do is I want to pivot our consideration of just the X chromosome and pivot to thinking about how the X and the Y chromosome interact and relate to each other. You already know that our sex chromosomes evolved from a pair of autosomes about 300 million years ago. This means that 300 million years ago, the X and Y participated in recombination, just like all our other chromosomes do. But 300 million years ago, the X and Y participated in recombination, just like all our other chromosomes do. But 300 million years ago, the X and Y participated in recombination, just like all our other chromosomes do. So But once genetic sex determination took hold as an evolutionary strategy, there was a major disadvantage to recombination. And over time, less and less of the regions on the sex chromosomes recombined. Today, the X chromosome recombines with only a very small part of the Y chromosome. Oops. My gosh. Oh, sorry, I forgot to remove all of the animations from my slides before I started recording. Anyway, so today, the X chromosome, it recombines with only a very, very small part of the Y chromosome, a region that we call the pseudo-autosomal region of the Y chromosome. Now we're going to talk about this region and so much more in the next video.